Development of the cochlear innovation. Thanks, Peter.
Thank you. So now this is a bit different to... can I get the slides up? No, I have to do it, right. I'm a little rat.
Yeah, and you explained it to us in great detail so we understand.
So actually, while we're waiting for the slides, just to let you know that for those people aren't aware, we've got the celebration of the 25th anniversary of the audiology program in Auckland on the 10th to 11th and 12th of September. And so hopefully a lot of you have already received emails about that, but I'm getting a lot of nodding heads.
Yeah, I know.
So it's going to be on in Auckland, and we've got a symposium about research, and there's a dinner on the Saturday night, there's a cocktail party on the Thursday night, dinner on the Friday night. So there's something coming out to everybody, hopefully within the next week. And so it would really be wonderful if you can all be there and enjoy just catching up with old friends and also just hearing a lot about some of the exciting research that's going on around the country. So what I'm going to talk about today is very different to probably what you've been listening to already. But one of the reasons for doing this is I want to talk about some of the work which we've been doing over quite a period of time, looking at aspects of how the innovation of the cochlea and the cochlear nucleus develops. And one of the reasons for talking about this is, as I say, I haven't really talked about it before, but we're really interested in trying to move some of this work into more clinical areas, or certainly thinking about it in terms of degenerative changes in the ear and in the cochlear nucleus. And we'd be really delighted to get more engagement with some of the clinicians who might be interested in doing research and being part of a multidisciplinary team looking at some of the whole processes about degeneration of the auditory nerve. So what this is about a program which we focused on the development of the nerve and the cochlear nucleus. So we're looking at rodents. We're mostly working with rats and mice. And as I said, a lot of this is actually thinking about some of the issues around developmental abnormalities. How do these actually develop and what might be the downstream consequences of that? In particular, we're thinking about what are the mechanisms and consequences of neurodegeneration inside the cochlea. So this idea, particularly in adults, of cochlear neuropathy, where neurons actually are degenerating as a consequence of, say, noise exposure and age-related changes, and we're not seeing necessarily that depicted in an audiogram. What is the consequence of that sort of process at more central levels? I can't cover all of the things, so I'm going to talk a lot more about some of the cochlear work which has been undertaken and just think about some of where that's going in terms of possible clinical work. So some of the questions we've got really is really around what is the sequence, what are the critical periods, what is the regulation of the normal development of the auditory nerve and how that connects with the sensory cells, and how it also then projects and forms the networks within the cochlear nucleus. And then thinking more about what happens if you perturb that during these critical periods or you perturb it during its development. For example, we've been doing some work looking at what's the impact of conductive loss. So if you produce a conductive loss in an animal, what happens to a lot of these changes actually in the central nervous system? What happens with perinatal hypoxia? So we've got some models at the moment looking at hypoxia and particularly extreme hypoxia and thinking about how that impacts on neural development. And these have really quite significant clinical implications, as you probably are aware. And as I said, we want to go and look at some of the impact of what we sort of term cochlear neuropathy, and which is sort of becoming a more substantive term in the literature, and look at how that impacts on central connections and processing in adults. And here we're thinking more around doing behavioural studies as well as electrophysiological studies to look at the processing problems. So the approaches that we have, these are all animal models. So we use mice and rats. We also have some work going on in guinea pigs. We use a lot of different techniques, anatomical techniques, molecular techniques. We have a number of electrophysiological processes, and particularly building a collaboration with some people in Otago to be able to do this in living animals as well as in vitro ones, which is where we're taking the tissue out of the ear or taking it out of taking slices of the brain, for example, and then studying the electrophysiological responses under in vitro places. We also have real-time imaging techniques to be able to look at how the cochlear tissue develops. You can take the tissue out of the ear, put it under a dish, in a dish, watch it developing, and measuring some of the molecular and structural changes that actually occur as the innovation and so forth occurs. We're building up techniques to look at behavioral assessments. But one of the other things which is really interesting is we're starting now to work with some adult human tissue, which is of the brain, which is coming from the brain bank at the University of Auckland, and starting to do some work looking at things like the relationship between changes in these tissues and conditions like Alzheimer's and Parkinson's and so forth. Now, all of this work is done by quite a lot of different people, and I just wanted to point out the work by, I'm going to talk a lot about the work by Megan, Lin Chen Huang, Cherry Mao, and I won't talk so much about Nishani's work, but these are people who have contributed enormously to the work I'm going to talk about. And I just want to also point out another group here with Courtney, Adelina, Tamina, and Charlotte Gordon, all audiology students who have also contributed to this work. So just, can I have the lights down a little bit? Please, thanks. So just thinking about the primary auditory synapse. I'm going to talk very much about the cochlea and rather than some of the central processes. So the area we're talking about really are the hair cells inside the cochlea, and we've really focused a lot on the innervation of the inner hair cells, not so much the outer hair cells, although I will talk a bit about that. And so this is the primary auditory sensory cell, as you know, I hope you know, and this is these neurons which innervate the inner hair cell, and there's a variety of different types of neuron which have different intensity profiles and different response properties. And what I'm going to show you is some pictures that look a little bit like this, and these little dots and things, they relate to the actual site. Here's the inner hair cell, and these red ones here are the synaptic proteins on the inside the inner hair cell, and the green ones are the glutamate receptors on the surface of the synaptic boutons, and I'll talk a bit about those as we go through. And so we're very interested in how does this innervation occur. So once the neurons actually come in and form a synapse with the inner hair cell, how do they get there? How does it form? And then how does it refine to actually become functional? And this is just showing you the adult form. So you see that there's neurons, about 20 to 20 neurons or so that innervate the inner hair cells, and the innervation to the outer hair cells is much, much more diverse, and one nerve fiber will innervate a large number of outer hair cells. And this is something taken from Lin Chen's PhD thesis, in which we've stained the type 2 fibers. So there's two types of fibers, the type 1s that are going to the inner hair cells, the type 2s to the outer hair cells. And the type 1s are in red, as you see here where the inner hair cell region is, and the type 2s are in green. And you can see that the majority of nerve fibers here are the spiral ganglion cells, are the type 1s compared with the type 2s, which are not so abundant. And another peculiarity or important thing about the hair cell is it has this thing called a ribbon synapse. So here's the hair cell at the base of the cell, here's the neuron, here's the bouton, and this structure here, this thing called a ribbon, you can see it actually in an electron microscope here, is a structure that enables, it's like a conveyor belt if you like, and all the vesicles containing glutamate are sort of attached to that, and they shuttle to the membrane. So you have this very, very rapid timing mechanism which will enable the release of neurotransmitter in response to a stimulus. And on the surface of the boutons here, the afferent fibers, are the receptors for the various types of neurotransmitter, in this case glutamate. So in the first studies which we've done, we've looked at and this is mostly by Lin Chen and Megan Barclay, and what the purpose of these studies was to look and ask the questions, what happens is that all of the neurons, all of the type 1 neurons actually innervate every hair cell in the cochlea at birth. And then over a period of time, they're refined until finally all the ones on the outer hair cells are the type 2s, and all the ones on the inner hair cells are the type 1s. So we've been interested in what actually happens in that process, and part of that was we found it had a technique which we developed, or Lin Chen developed, I should say, which enables us to stain in real time the type 2 fibers, which are the green ones, and the type 1 fibers, which are the red ones. And what you see, this is before birth, and you see that the fibers just look quite disorganized. There's just a whole array of nerve fibers out to the hair cells, not a particularly ordered appearance. And if you look in a mouse between the ages of soon after birth to about 6 days after birth, there's a total refinement. So you see this sort of disorganized appearance until finally, even within 6 days, it's become very, very highly organized. And so what you see is the type 2s here, which are going to the outer hair cells. The outer hair cells have a lot of type 1s to start with, and over a period of 2 or 3 days, they're just retracted until finally you've got this firmed up innervation of the inner hair cell. And this just shows that maybe a little bit more clearly, but you can see here the type 1s going out to the outer hair cells, and after 6 days, they've retracted, so it's only on the inner hair cell fibers here, or cells, and on the type 2s, you see that they're innervating the outer hair cells, and that's consistent throughout. One thing you just might notice, actually, by the way, is that some of the type 2 fibers actually also make connections with neurons beneath the type 1s. Now, this is probably very hard to see because of just with the lighting, but all I want to just make a point here is that what Lin Chen and Megan have done is looked at a lot of the molecular changes, trying to work out what actually goes on as these fibers retract and refine. But this is just showing, these are the number of, you can see these little red dots. Here's the outer hair cell region, here's the inner hair cell, and the little red dots are where the synapses are. And so when these nerve fibers actually make a contact with the outer hair cell, they form synapses. They're proper synapses. But then something happens, the cells don't respond. So this cell, basically the nerve fiber says, well, if you're not going to stimulate me, I'm going to bugger off. And so it retracts, and it retracts very, very quickly away from that fiber. But that is a fiber that has actually come, it's also connecting the inner hair cell, it's connecting the outer hair cell and the inner hair cell. It just retracts that one dendrite, which is fitting the outer hair cell. And so you see by the age of 6 days here, you've got all these, it settles down to the number of synapses at the inner hair cell, very large number to start with, and then it prunes to have just a small number, and they're pretty well gone in the outer hair cell by 6 days after, or beyond 6 days. And this is just showing you, here's 3 outer hair cells, there's a whole lot at P3, you can see all these little dots, and there's very, very few by P12. And this just shows you that these neurons are just disappearing after 6 days. And we're able to, if you correlate this with the, this is probably too difficult now to see because of the color, I'm sorry, but if you do a comparison between the ribbon, which is the protein we're able to focus on here in red, and the synapses, the synaptic proteins, the glutamate receptors in green, you can look at the relationship between these two things under the microscope, and you can see that the outer hair cell does form proper synapses, but because they don't appear to be functional, that's when they disappear. And you only find that these, and it's very hard to see here, but you only find the ribbon, it's only when the ribbon and the glutamate receptors are actually opposing each other will it form a proper synapse. So you end up with a lot of ribbons inside the cell, a lot of synaptic proteins inside the cell, which then actually don't find themselves against a glutamate receptor block, so therefore they just disappear because there's no functional connection. So you have to have this correlation between the two for it to form. So from a lot of work, the summary we can come to is that if you look at the inner hair cells and the outer hair cells, at birth, every cell receives all the type 1 neurons. Everything goes to pretty well every cell. And over the period of a few days, the synapses start to diminish in the outer hair cells we see here, and you see this process in which the nerve fibers just retract. And the synapse that's left forms a proper synapse with the type 2s, and that becomes mature, and the same thing occurs by P12, or about 12 days after birth and into the adult. You've seen this whole thing firming up. Certain proteins now become expressed, and these neurons actually are now have the ability to be activated. And so this process seems to have this process of outgrowth, where it forms the synapses. It then has a period of refinement and then retraction, and that whole process occurs in the mouse anyway over a period of about 4 or 5 days. So what about, how does that relate to hearing? What does this mean in terms of how the animal hears? Well, this is some work that Courtney Manderson did for her master's thesis, in which she actually looked at the onset of ABR, using ABR in this case, to look at the onset of hearing. And this is a mouse at P8, and at P8, even though these neurons have actually established, these animals don't hear. There's no response. And so if you look at the audiograms from the ABR, you see that over a period of time, they get more and more sensitive, and we call this period in which it gets sensitive over a period between about P12 and P15, the onset of hearing. It's way, way after the time in which the neurons have actually established. So even though the neurons have actually established, the cells we know are actually activated, there's no synchronized impact, or there's no hearing, if you like, from those neurons. So the question is sort of quite interesting then, is, well, if these neurons are not being stimulated by sound, what keeps them active? What makes them function? If the neuron needs activity to maintain the connection, how does it do that? If there's no sound coming in, if there's no activity. Well, what's interesting is that in the period prior to the onset of hearing, the hair cells become active. The inner hair cell in particular starts bursting with spontaneous neural activity, and it stimulates the neurons that then sends messages to the brain. And we believe that those messages then, and there's a lot of work now that shows that those messages are critical for setting the pattern all the way up to the cortex. And the organ of corti bursts in this way, the spontaneous activity differentially along its length, so that the basal turn and the apical turn are doing it differentially, so it's actually forming a tonotopic pattern all the way through to the cortex. And so that bursting of activity is essential, the spontaneous activity is essential for setting the relationship so that when the sound and the ear opens and it can now become active with airborne sound, this spontaneous activity turns off or diminishes, and the sound-mediated activity increases, as you saw, with that increase of threshold. Yeah. These are days after birth, sorry, yes. So the other one, yes, so these are when it's born. So this is sort of getting into probably 30-week sort of gestation, if you think about it in terms of a human. And so this spontaneous activity is now driving it. And Nishani Dariatni, who's one of our PhD students, has been studying a lot how this spontaneous activity is actually generated to produce this patterning inside the central nervous system. So I just want to talk about Megan's work. She'd be horrified, there's such a big picture of her. She's been doing a lot of work in this area and is a postdoc with us now and has her own grants and is developing this area. And if people are interested in working, she's the person to come and work with. But one of the questions she asked was, well, if that sound is important, if the sound comes in at that later stage, is it important for actually maintaining and influencing the maturation of the synapses inside the cochlea? And so what she did was to produce a conductive hearing loss in these animals at about P11, just before the onset of hearing. So we just actually dislocate the chain, all sorts of problems in trying to do this, but we just dislocate the chain, or she does, I should say. And then we can study the auditory brain responses at different times after birth, and then look at taking the tissue for microscopy. And if you look at with the control, this is the auditory brainstem response in these animals. This is by age 21, P21. And after dislocation, you can see there's quite a big threshold shift. You see the response is only coming in around about 40 to 45 decibels. And if you do an audiogram using the ABR, you can see here that there's a dislocation. This dislocation produces a conductive loss of around about 30 to 40, maybe up to 50 dB in some cases. And I'll just go through these data, but what's quite interesting is that what she found was if you look at these are the ribbons. So these are the actual number of synaptic protein sites inside the inner hair cell. And if you look at the different ages and we look at the dislocated ones, you probably can't really see much of a difference. You might not be able to see anything at all because it's so dark. But she's quantified all of these, and what you find is that the number of synapses doesn't change. But what's quite interesting is that, and it might be difficult also to see this, but the size of the synapse changes. So these animals seem to have actually been adapted to this lack of input. And if you look at the number of receptors, the glutamate receptors, the same thing happens, is that the number of receptors doesn't change, which is what you might imagine, but the size of the area, which has actually got receptors in it, increases. So you see this bigger distribution, this bigger distribution. You see here, the dislocated ones go right out, very, very large volumes, very large size of these number of receptors. So what it suggests is that in all of this is that the aceticular dislocation gives rise to this conductive loss. It doesn't seem to change the number of synapses, but what it does seem is if it actually changes the amount of receptor, the number of receptors which are available, and the size of these receptors, which suggests that the cochlea can adapt. The synapse can actually adapt to the lack of sound, and since you've got this loss of input, it seems as if perhaps we think it's actually adapting to increase the probability of a response by increasing the amount of the synaptic proteins. So even when you've got this loss of this conductive loss, the cochlea does seem to be able to adapt to that change in the inputs. So just coming up to the end, the next things which we're thinking about, and some of the work which is going on at the moment, just give you a little bit of a taste of it, is thinking about the impact of hypoxia on the cochlear nucleus and some of the auditory processing. And we started off doing this thinking whether it had any effect on the cochlea if you made these animals hypoxic. So these are just after birth, when they're P0, just born, or even prior to that, we make them hypoxic, quite severely hypoxic, and then it's sort of almost mimicking what might be happening at birth with asphyxia. And what you see is that this is an ABR in a number of animals. You see the wave 1 and the variety of waves here. And what you see in the animals that are made hypoxic, there's no change to the early waves, but you see all this distorted morphology in some of the later waves. And Cherry Mao, who did this for her honours project, she did some calculations and showed that there's a big change in latencies and quite a change in some of the later waves, suggesting that the hypoxia is having an effect on the central processes in the auditory system, particularly around the cochlear nucleus. So what we've then gone on to do, and what Megan's doing now, is looking more at what happens if you, inside the cochlear nucleus, and with severe hypoxia. And she's only these are very preliminary data, but this is just, by the way, is an image from Adlena's work when she did her master's with us last year. And you can see here, this is just the cochlear nucleus, the dorsal cochlear nucleus and the ventral cochlear nucleus in the mouse. And what Megan's just started doing is analysing, first of all, the number of neurons which exist in here before and after hypoxia. And what we see, which is kind of interesting, is that there's a reduction in the volume and the number of neurons inside the ventral cochlear nucleus and no change to the dorsal cochlear nucleus. So hypoxia certainly seems to be having an effect, and we're now going on to look at what the consequences are of that and what neurons are actually being affected. The other thing which we're doing is looking at, with the development of the dorsal cochlear nucleus, and then looking at what happens if we remove or change the cell population by destroying various neurons inside the cochlea. What happens to the development of the dorsal cochlear nucleus with peripheral neurodegeneration? So that's some work which is just on its way now. Cherry Mao has done a huge amount of work looking at how the cochlear nucleus develops in the mouse, and both physiologically and morphologically. We're now taking the next step, which is to look at what happens if you perturb some of these things and remove some of the peripheral neurons, and what happens to the development of the nucleus. And the other thing which Megan is looking at is I mentioned that there are different populations of neuron inside the hair cell, so attached to the hair cell. They have different response properties. And the question is, well, how do those develop? Because it appears that they all start off the same and then they become different over time. And so she's just having a look at some work at that. So that's another project. And there was a final one which I was going to talk about, but I think we'll just leave it at that. And I put this here because it looks like a question mark, so I thought it's reasonable. Okay, thanks. Highway.
Is there any way that you can look at stimulating it so that the dissociation doesn't occur and actually see what happens to the cochlea?
This is with so you could just...
So if you're saying that those ribbons dissociate.
Right, yeah.
Getting in there and stimulating before they dissociate.
Yeah, it's a really interesting idea. And one of the ideas we had was to use essentially like a small cochlear implant. And so you stimulate it. One of the problems that these animals are very young, the cochlea is very hard, was actually very soft, but it's very difficult to get into and to do that. So what we were thinking was actually just putting a sort of a big ball electrical stimulation sort of close by and just stimulating it. See if you can actually maintain those. It's a really good question. It's quite hard to do that, but if you could just do sort of a blanket electrical stimulation, would they actually would they retract? Would be a really interesting question, because I mean these things, and from a developmental point of view, what actually governs all of these processes, but what happens if they don't happen? So what happens in a child, for example, where some of these processes might not actually happen, whether these changes don't occur in the appropriate developmental stages? What happens to the inputs to the central nervous system? And that's the sort of question that you could answer with that sort of approach. Yeah.
So thinking about that in a baby, if their ear canals were blocked post-birth, it's the same thing, isn't it? If they stayed that way, what might happen once that was cleared in mice? If you put them back.
Yeah. Yeah. We don't know that at this stage. I mean, I think the first thing is that what it does appear, and I mean, how you translate this into the clinical situation, which is why we'd like to get some help with that. I think that our view in the cochlea is that the innervation actually adapts. So it adapts in terms of upregulation and downregulation. And so if you, for example, look at animals which have been exposed to background noise, so just in adults, just background noise, the number of receptors actually drops. So it's adapting to its environment. It doesn't need all that. And when you take that off, it comes back again. So these are quite responsive. So perhaps, and in an adult, it might be different because it's actually, it hasn't fixed. It is fixed, but it might be able to adapt. What happens with these animals if you then turn it off and then you put it back some time later, whether it comes back again, we don't know. But my view is that the cochlea is very adaptive, and I think that's something that's quite interesting with a lot of the issues around this sort of continual decline of neurons. Is whether those adaptive mechanisms are actually not at play, and so that becomes vulnerable to injury. Johnny.
Is there any ideas or suggestion about how this sort of reorganisation might be occurring in human infants? Is it pre-birth? I mean, I kind of imagine it probably would be, but
yeah, it would be. Yeah.
And I suppose the implications of that is we all think they're probably hearing in the second trimester or something, isn't it? But it's sort of an impoverished stimulation compared to obviously ear conduction, if you want to call it that. Any thoughts or anything?
Well, I mean, I think it's hard to. One of the difficulties with this is how you actually do relate this back to humans, because you've got a precocious environment compared with this sort of altricial environment. So it's quite difficult. But there is. I mean, I think probably I don't see why the processes shouldn't be the same. And what's really interesting is those spontaneous mechanisms occurring, or if you've got in utero hypoxia or things that are happening at critical times, perhaps some of those things don't occur in the right sequence or at the right time. So I think this is actually also not about whether I think you'd be looking at more complex processing than just perhaps detecting thresholds. So threshold is what you can measure, but in terms of actual processing, what's going on in the central nervous system if you don't have all of these events occurring, because it's quite critical. I mean, the spontaneous activity is really fascinating. I mean, it's handshaking between the ear and the brain, and if that's not occurring in the right sequence and in the right way, then do you get normal pattern development inside the central auditory system, and how does that play out further down the line? It's really interesting. Yeah.